Okay, well, I know a few of you are still getting your lunch, so don't worry, take your time. But we're going to go ahead and get started with today's program. We know that living with ALS or caring for someone who is can feel overwhelming at times. There are appointments to manage, decisions to make, and challenges that often seem to change from day to day. But that's exactly why we gather for events like this. Our hope is that today's Lunch and Learn provides not only valuable information, but also a reminder to you that you are part of a community that understands, supports, and walks alongside of you every step of the way. Before we begin, I'd like to thank our volunteers for helping us today, and we have some board members here that support us throughout the year as well, so I thank you all so much. And I want to introduce to you our incredible staff team as well. Our staff is dedicated to serving individuals and families affected by ALS throughout Georgia. While each of us have a different role, we all share the same goal, to ensure that no one faces ALS alone. So I'd like to ask our team to stand and wave when I introduce you. So you all have to kind of look around the room. Most of us are, I think all of us are in our light blue attire of some kind. So Liza Nordmark, our Senior Director of Philanthropy, she's up here in the front. And her team, Ellen Strickland, is our Senior Donor Relations Manager. Many of you know her. Allison Bailey, our Walk Coordinator. Caitlin Wolf, the newest team member with us, Communications and Development Coordinator. Our Care Services team, Anita Flowers, Senior Care Services Program Manager. Robin Rakoff, our Care Services Coordinator, focuses mostly on Central and South Georgia and Northwest Georgia areas. Stephanie Medill, our Care Services Coordinator, Metro Atlanta area mostly. Amanda Fiesler, who could not be with us today, is also a Care Services Coordinator, and she resides in Augusta and works with our Northeast and Coastal Georgia clients. So Pam Amy Cupp, our Administrator Coordinator, who's back here in the back. She keeps us all afloat in some way, shape, or form, whatever we need, so we're always grateful for her. And I'm the Executive Director, Sarah Imbro, and I have the privilege of working alongside this incredible team. At ALS United of Georgia, our mission is simple but deeply meaningful: to enhance the lives of Georgians living with ALS and their families by providing personalized care services, fostering a supportive community, and advancing research focused on treatments and a cure. Everything we do is centered around improving quality of life while offering hope for the future. Many people first come to us looking for information, but they quickly discover that we're much more than a resource. We're a partner throughout the ALS journey. Thanks to the generosity of our donors and community supporters, our programs and services are provided at no cost to the families we serve. Some of the ways that we support you and the ALS community include, as you can see up here, personalized care services through our experienced care services team, equipment and technology device loaners through our Ricky P. Sweat Equipment and Technology Assistive Technology Program. That's kind of a mouthful. That helps you remain safe, comfortable, and independent. The Sally Panfell In-Home Care and Respite Program. This helps provide much-needed relief for caregivers and additional support in the home. Our Paul B. Williams Transportation Program that offers transportation assistance to families to access ALS clinics, medical appointments, and other essential services. I believe we even had a few people use that program today to be able to attend today's luncheon, so we're glad you could be here. Educational programs like today's Lunch and Learn, support groups, caregiver resources, and other opportunities to connect with other people who understand the ALS journey. We also do advocacy work as well, both at the state level and the federal level, working with other ALS organizations to give a voice to the ALS community and to work towards policies that improve access to care. And finally, the highlight of our program today, we support research initiatives that bring us closer to better treatments and ultimately a world without ALS. Today's program is just one example of our commitment to keeping you informed, connected, and empowered. So we encourage you to ask questions, meet our staff, connect with one another, and take advantage of the resources available to you. If there's something you need and you're not sure where to turn, please ask us. If we don't have the answer, we'll be sure to help you find it. Before we continue, I would like to invite Liza to come up here and talk for a few minutes. One of the things that makes us so special is the incredible network of supporters that help make our mission possible. So Liza's going to recognize some of those partners and share a few exciting updates for you as well.
Okay. Yes, thank you all for being here today. And before we talk about some upcoming events, we definitely want to thank our community partners who are here. We're so grateful for you coming out. So many of you come out every year, and I know that it means a lot to the families. So we have, and would like to thank Shinogi, Pulmonary Care Services, New Motion, R&R Mobility, Agape Hospice Care, Accessible Living Atlanta, Toby Dynavox, and Kate Barnes. You just click it. Great. I'm also excited to share a little bit about Walk ALS with you today. It will be on Saturday, September 19th at Center Park Stadium, which is the old Turner Field. It's changed their name so many times. I know it's hard to keep up, but this year's walk is especially meaningful. It is the 25th anniversary of Walk ALS here in Georgia. And that is such an incredible milestone. It's incredible for an event to be around that long. It's incredible for an event to raise as much money as we have together. And it's really something that we're looking forward to celebrating with everybody. For 25 years, families living with ALS, caregivers, families, friends, and supporters from all across the state have come together to walk side by side, to connect, to celebrate, to remember, and to raise funds that are critical for our programs and services that Sarah just spoke to you about. Walk Day is so much more than an event. It is a day where we come together as a community. On my team, we say it's our Super Bowl. Because it is. And it's a day to celebrate those we love, to honor and remember those who are no longer with us, to connect with others who are in the community, and to be surrounded by those who want to show their support. Teams in matching shirts, families and friends gathering together, music, face painting, other activities, and team spirit everywhere. Walk Day is a day of celebration, and it is a day of hope. I know that some of you here today already have walk teams, so yay. And some of you have been walking with us for years. For those of you who maybe have not been and would like to have some more information, please come see us over at the walk table. Allison and Caitlin are there. And we would be happy to give you some more information. Wherever you are, we hope you'll be a part of this special anniversary year with us.
Okay, so now it's time for the learn part of the Lunch and Learn. So I'm going to invite our guests, our keynote speakers up while I make introductions of them. So most of you may have seen one of these doctors in a patient room, but I bet you didn't know all of what else they do. So I'm going to tell you a little bit about that. Dr. Christina Fournier is an associate professor of neurology and clinical researcher at Emory University and the Atlanta VA Medical Center. She serves as co-director of the Emory ALS Center and director of the ALS and neuromuscular programs at the Atlanta VA Medical Center and director of the neuromuscular division at Emory. Dr. Fournier's research focuses on strengthening ALS clinical trials and developing improved tools to measure disease progression, with the goal of accelerating the development of effective treatments. She has led and participated in numerous multi-center ALS clinical trials, served as principal investigator on Department of Defense and VA-funded research to advance ALS outcome measures, and contributes to national initiatives shaping the future of ALS clinical research. Dr. Eleanor Thomas is a neurologist and physician scientist at the Emory ALS Center and assistant professor of neurology at Emory University. She specializes in the care of patients with ALS and other neuromuscular disorders. Her research focuses on developing blood-based biomarkers to improve ALS diagnosis and prognostication using high sensitivity proteomics and machine learning. I think I said that right. Dr. Thomas completed her MD PhD at Yale University, followed by neurology residency and neuromuscular fellowship training at Emory University. She is the recipient of the Richard Olney Clinician Scientist Development Award and from the ALS Association and the American Academy of Neurology. I know you already knew they were smart, but I bet you didn't know they did all of that I just mentioned and probably didn't pronounce very well. So we appreciate their willingness to share their expertise and time with us outside of the patient room. I'm going to turn this over to them now. You all have note cards on your table, so if you have questions while they're speaking, feel free to write them down. And at the end, when they're done talking, you can raise your note cards up and we'll work the room and collect those cards for the question and answer session we'll have when they're finished.
Very good. Well, thanks for being here today. It's great to see a lot of familiar faces. We're going to leave plenty of time for questions, but we wanted to talk a little bit about the research approach we have at Emory, which is we find it really meaningful to make sure that we learn from every single person with ALS that comes into the clinic. So many of you were asked when you first met us if we could study your information, your blood, for some people, your spinal fluid, your skin, and learn everything that we can about ALS from you. And we've been so impressed with the generosity of our patients in this joint mission to learn from every person living with ALS and then bring that to our research program. So you probably know that the history of ALS research and kind of learning from a mouse has been very limited. We have a lot of treatments that work great in a mouse with ALS that have not worked in people. So we think an approach that we need to do is to actually learn from people living with ALS, take that back to the lab, and then bring treatments forward. And that's what we're going to talk about a little bit today, kind of what we've learned so far and where we're hoping to go with our research program. So we, for decades now, have had what we call lovingly CRIALS, which is the clinical research in ALS studies. So this is where every person who walks in the door has the ability to contribute to ALS research and help us learn more about this disease. And so this is just a little slide that shows you where we have, each participant is from, obviously centered around Georgia, but really people all over the country. And the graphs that kind of describe who's in this just show that unlike some natural history studies that only get a small sample, we're really able to capture a representative group of people with ALS, which is really important because if your ALS is different than your ALS and there's different biology there, then we need to be studying every kind of ALS to figure out treatments. If we do end up with a precision approach where, like the gene therapy that we have now that only targets a specific gene, then we need to understand the entire ALS population. And so we have this really rich resource that you guys have given us where we can study all of the different types of ALS. So just to give you an idea of what you guys have given us. So we've enrolled over 250 new participants since 2024. We have thousands of patients that have contributed their clinical data. We have blood samples. We have spinal fluid. We have what's called whole genome sequencing, which is this really rich genetic information. Some who have even donated brain and spinal cord afterlife, and that gives us a really rich resource to study ALS biology. So we have this huge resource that we take very seriously that we must learn from this to come up with ALS treatments. And this is all thanks to you guys who have been so generous about it. Certainly don't need to look too hard into the slide, but this is just a sampling of some of the papers that have come out of this data set. So just to give you an idea of the kinds of things we've learned already. So we've learned new ALS genetic information, risk genes, and genes that contribute to ALS, and that gives us a clue about biology and potential treatments. We've learned about biomarkers, things we can study in blood that tell us about ALS prognosis or ALS biology that could help in diagnosis. We've learned a lot about ways to track progression in ALS, which is really important when you're doing research to see if a drug is working or not. We've learned about disparities in care and in diagnosis so that we can focus on clinical efforts that do better in diagnosing and treating ALS. And we've learned a lot that tells us, that allows us to predict progression and predict disease traits over time, which we'll talk about a little later, one application for that. Okay, so I know it's a Saturday morning. I will not bore you too much with math, but Sarah mentioned that one of the things that I'm very passionate about is how we track ALS progression. And the reason for that is that it's really important to have good tracking tools if we're going to do good clinical trials. So what you're looking at here is probably everyone with ALS in this room has completed what we call the ALS functional rating scale. So this is a scale that asks about your speech and swallowing and your arm movement and your leg movement. And many of you have been very generous in completing many, many scales. And each little line here represents the progression of an individual patient. And what you can see is it's very, very variable. Just like each of you in this room looks different, everyone's progression can look a little bit different. And so I want you to imagine, since this is the outcome measure that we use in a lot of our trials, what if you had a drug that maybe slows progression by like 20 or 30%? Or what if you had a drug that works in maybe 20 to 30% of people, and you're trying to see that signal through all of this variability. And you can imagine that is tricky. So any way that we can track progression in a more robust way, in a more reliable way, that's going to detect changes in a more sensitive way is going to actually help us do better trials. And so to me, this is very low-hanging fruit that if we can measure ALS in a better way, and that leads to faster trials, to trials that have a clearer result, like this is very, very important and something that we should be doing. And so that has been, like I said, through a lot of your generosity in completing a lot of these outcome measures, something we've made progress on. So many of you who have also completed this road scale, which we created at Emory a few years ago, and now it's used worldwide and used in trials. And the idea with this is in that ALS functional rating scale that we talked about, sometimes a point can mean like a little bit of functional change, and sometimes a point can mean a big amount of functional change. And so when you're looking at the total score and you're trying to measure, does this mean, what does the score mean? Mathematically, it has problems. So the roads, again, not too much math, but it has these mathematic tools that actually allow you to measure change in a very consistent way. So when you look at the score, it's more meaningful. And so we've been able to roll this out through you guys, validate it, and like I said, now it's used worldwide in trials. Another thing we've been able to do is when people come in, we ask them, hey, compared to last time, like, do you feel like you're a little worse, a lot worse? Like, you tell us the amount of change that you've had. And then we've been able to describe what's clinically meaningful change according to you. And that hadn't been done before. And that I think is really important when you might be considering a new treatment and you want to know the effects. We can tell you, well, according to people living with ALS, this is a meaningful amount of change. It shouldn't be something we define. It should be something that you guys define. And we've been able to characterize that through some of our research. Some forward-thinking things here. So now we have a lot of digital tracking tools where we can learn about your ALS without you ever coming in. So there's an ongoing study now. We partnered with this group, Everything ALS, who's very focused on tech and ALS, and they used a scale that we also developed at Emory that assesses motor function. And so when we initially looked at the scale, it was during COVID, we were doing telemedicine visits and tracking motor function, and learned that we were able to do that through telemedicine visits, but now it's like through a self-guided app where the app will show you these motor movements and describe them, and you can rate it, and then it'll track progression over time. So one exciting direction that we kind of want to put this all together and as an upcoming project is that we want to use these ways of tracking at home and these prediction tools that we have for personalized education. So there's a lot of educational stuff out there, a lot on the internet, and it can be very overwhelming, especially like at that first visit where there's so much to learn and it can be tricky to figure out when to tell people things when. So the idea would be that you would input your data in your smartphone, and these prediction models would say, okay, this is the educational module that is useful to you right now. Like, we predict that maybe you need some mobility help soon, so here's education about that. And it would roll out these educational modules based on your predicted progression, and then it would ping us as well and say, hey, we just determine that this is a relevant module so you can follow up in clinic. So this is an early project, still working on funding it, but I think one way that we could take all of this data that we have and really improve the care that we give. So we're excited about where this could take us. And now I'm going to let Eleanor talk a little bit more about, I might have to hit it for you, what's going on in the lab.
So one application, so what, Dr. Fournier also already told you about Cryos, where we're collecting blood from all patients that want to participate that come in our clinic. And we have stored that blood for now, a couple decades, and we're beginning to study this in kind of robust, quantitative ways to see what proteins are different in patients that have ALS. How do these change over time? And are there ways that we can, from a simple blood test, diagnose ALS earlier and also integrate that with some of the clinical data that Dr. Fournier told you about in order to come up with better prediction algorithms. So the first thing we did with this blood is we tried to address the problem of diagnostic delay, which probably a huge percentage of people in this room have observed firsthand, either in themselves or a family member, where just going to see doctors, sometimes the symptoms aren't recognized as ALS, and if these could be recognized earlier, it could help, patients could get better supportive care earlier, have improved access to physicians specialized in ALS, have better access to research, have better access to trials. So I studied diagnostic delay in patients in our clinic. So how much of a delay is there? So the average person isn't diagnosed with ALS until 17 months after they recognized a change. So when we say, what is the date of onset, when did your symptoms start? That's what you report, and then when were they actually diagnosed, either by us or another neurologist. So that's over a year and a half, or almost a year and a half. And then if you kind of look at this in terms of what is the overall disease duration in a patient, because some patients may have a disease duration of a short period of time, some patients may live many years with this, and so if somebody's had ALS symptoms for 3 years, which is a longer time period, then maybe if they're being diagnosed later, maybe they're just a slower progressor, for example. So if you look at where somebody is in their overall illness, almost 30% of people are halfway in before they've ever even been told that they have ALS. And the problems for that are, there are many. One is there just isn't enough neurologists. The neurologists we have don't have sufficient training in ALS, so maybe even though they should be able to recognize it, it's difficult. ALS presents in many ways. People have different patterns of weakness. One person may have facial weakness, another person may have weakness in their left hand, and even though those look very different, like different diseases, they're one disease. And so all of these things lead to delays in referral to neurologists, then another delay in referral to specialized ALS center, less access to supportive care, whether somebody needs to address breathing problems, nutrition, mobility. All these things don't happen until someone has a diagnosis, oftentimes. And if we look at referral patterns, most people see their primary care doctor first, which makes a lot of sense. Only a couple percent go to a neurologist as their first visit. And after seeing a primary care doctor, the people are referred to neurology, but also neurosurgeons, ENTs. We had a couple patients that saw a dentist first, orthopedists. And all of this is just kind of waste time. It may even lead to unnecessary procedures, wrong diagnoses, and I think some more suffering than is necessary. So we need better tools. I hope I've convinced you of that. And you can imagine that just being able to measure something from the blood is kind of a clinically sort of practical, because everyone gives their blood for a bunch of different things. You go to a doctor's office, you're going to get blood, And it doesn't require any specialized knowledge or training in order to measure, to obtain blood. And this is something that an orthopedist could also measure blood just as well as a primary care doctor could, and could be used in rural areas, cities. It really doesn't matter because it's blood. And if we could come up with use the data that we have from proteins that we've measured in blood to come up with a test that could triage people at very high risk of ALS or low risk, give some sort of score. And then we would see that, oh, this person got a high score. They need to be seen in the next month. We need to see them right away, for example. That would help us too. The tools have really improved for measuring proteins in blood over the past 5 years. I mean, very recent. And everybody has thousands of different proteins in their blood, but most of those are things we don't care about, like albumin or just proteins that everybody has that aren't really different from individual to individual. And the ones we really care about are probably really rare. So neuronal proteins. There probably aren't a bunch of proteins from neurons in the blood of anyone. So we need something really, a very sensitive assay to measure what we care about. So this is one platform, and there are a few very similar platforms, but this is the one we're using, where it's using antibodies and antibody pairs, so you need to have 2 antibodies that can recognize the proteins of interest and then amplify that signal, and then we get a quantitative result that we can then, and we can measure them simultaneously. So 350 proteins at once, and some platforms can even measure quite a lot more than that. But a few hundred ends up being kind of the sweet spot of being very accurate and sensitive. So we took that data and we put it in a few different machine learning programs, because you can imagine it's very complex data. If you have data from 500 individuals, each of them have 350 proteins apiece, and then you add clinical data, now you have a lot of different things that you're measuring at once, and you want to be able to filter that information to inform whatever question you're asking. So the question we asked is, can we take all of our control blood samples, and we had a couple hundred of those, and classify those as control accurately, and then classify all the ALS samples as ALS accurately. And how many proteins do we need to do that? Do we need all 300, or is 1 enough? Is 10 enough? And how accurate can we be? So we tried it. There are a lot of different algorithms for doing that, but we found one that was able to classify the ALS samples from control samples with pretty like 99% accuracy, pretty much. And then neurofilament's a protein you may have heard about. We do measure this, and it's an important ALS biomarker. But if we add 9 more proteins to this, we improve accuracy quite a lot. And if you're going to apply something in a primary care setting, you have to be very accurate. So I think that's an exciting future direction. We're looking to test that more. So when we're collecting blood in the clinic, we're actually saying, testing these 10 proteins. Are these the right 10? Do we need to add more? Do we need to take some off to really refine that? And hopefully that could be a clinical tool that would help people, I hope, soon. The future, where we'd really like to go is now we can say what's ALS in control. Can we find different clusters of ALS? Or do different patients have different ALS phenotypes? And can we estimate progression with these blood proteins we're measuring? So that's a little bit about the research. So it's really samples from you that we research, and now we're going to try and take those back to the clinic and help patients.
We wanted to leave plenty of time for questions, so.
Okay, so that was a snapshot into some of the work they do behind the scenes at the Emory ALS Center and the research lab. And they also brought some members from the research team. And there's a table up front. If you haven't talked to them, you can see them before you leave too. If you all have questions, go ahead. Now's the time to start writing them down or raise them up, and our team will grab them. We have a few to start out with, so that gives you some time. And any question, for the most part, is a go, as long as it's not a personal medical question for yourself. You can save that for your visit. So we're going to start on some research topic questions. And we did ask for questions ahead of time, so that's how we came prepared. So, what is the screening process for clinical trials at Emory? Are you screening at an ongoing basis there, and are there age limitations?
Yeah, so every individual clinical trial has its own specific criteria that's determined by the sponsor of the trial, which is usually a drug company, and it's very specific to that trial. So there will be it varies from trial to trial. For example, a specific gene therapy trial might only be looking for people with a specific genetic cause of ALS. There might be a trial that is targeting inflammation, and you might have to have certain inflammatory markers. But each trial is going to have its own criteria for age, for duration of disease, meaning how long you've had symptoms. Some are safety things, like you have to have a certain vital capacity or breathing measurement. And then there can be individual safety things. If one drug might affect the liver, maybe you have to have no history of liver disease. So it's different trial to trial. One general theme that I know is frustrating for patients is that kind of back to that, if you want to measure change in ALS, it helps if someone is expected to get worse or progress over the trial period. Because if you think about you're testing a drug that is going to change the curve, slow progression, and in that 6-month period, someone's not expected to progress at all, you wouldn't be able to measure change even if it worked. So sometimes, for a statistical reason, there's actually criteria about how slow or fast someone is progressing. Biologically, also, a lot of these drugs, we expect they would be more effective early in disease, so a lot of times they're looking for earlier stage patients. So those are kind of some general things, but it is specific trial to trial. Everyone who's been in our database and consented to be contacted, when we have a new trial, we'll run the specific criteria, and then we reach out to people that are expected to qualify based on that trial specific.
How many active clinical trials is Emory hosting currently?
Right now, there are 2 recruiting and 2 about to recruit, and then there's one that is done recruiting but still active. But that number fluctuates just depending on
the timing. Right.
Yes.
Okay. Can you tell us a little bit about the Emory ALS research team? How large is it? How many are on the team?
Sure. And it's a big team, and what I think is exciting is it's not just the ALS team. It's kind of just like you've been in the interdisciplinary clinic, but on the research side, there's a lot of that as well. We have people in genetics, people in biochemistry, people in bioinformatics that collaborate with us in ALS. And so on the clinical side, we have, I'd have to count. We have multiple research coordinators and research nurses, and our physicians that work together. On the lab side, I'll let Dr. Thomas tell you a little bit.
I was trying to run through the numbers as we're talking. So probably, we have a full room for probably over 10 on the clinical research side, probably more than that. And then in the lab, 2 technicians, a couple students, 4 or 5 PIs that we collaborate with regularly. So I would say on the research side, it's at least 20, but that number kind of ebbs and flows depending on how many students are in the lab, funding. But from the research standpoint, it's 4 or 5 main investigators that will interface with samples, specimens, the informatic analysis and proteomic analysis, and then all the people that work for them.
Yeah, and I think one thing that makes it makes it unique and that I think is a strength is that it's not like the lab side and the clinical side, but it's very much integrated. So we're giving the clinical data and the samples from the clinic to the lab. We have new people in the lab will come rotate in the clinic to kind of get the big picture, like, hey, these proteins you've been slugging over, like, this is why it's important. This is why we need treatment. So It really is a big team that works well together.
Yeah, and I'll add to that. I think that's one reason why I really appreciate you allowing students in the clinic, because it lets them see this disease and really understand what they're working on.
Great. How does Emory choose which studies you'll participate in, and what stops you from being able to do more research?
Yes, so we always vet the science. As you know, we never are sure if a trial is going to work or not, but we want to understand the scientific rationale, believe that it has enough support for the safety and for the potential of this mechanism being relevant in ALS to do it. Then there's the logistics and the funding, which has been very tricky lately. This has been a tough couple of years for academic research and funding and federal funding for what we do. So unfortunately, sometimes we have to say no to good research because we just don't have the resources. But we do our best with what we have, and we have a really good team that works quite hard.
We do. The limitations are man hours and paying for those man hours. I would say is one big one. But with that being said, we've had enough, I think, to keep a robust program going through a difficult time, which it can't be said everywhere.
Okay, we're going to continue on and hope we are louder than the rain out there. For those of us with the C9orf72 mutation, or the genetic mutation, what research, clinical trials, or emerging therapies are you most excited about over the next 3 to 5 years?
That's a great question. I don't know of any specific trials that are about to happen, but I know there's a lot of interest in and revisiting the gene sort of therapy and gene editing sort of approach for that. So one idea is that everybody's DNA has 2 lines. There's it goes right to left or left to right, or you can think about it that way. We call that sense or antisense. So the gene therapy that failed in trial only looked at one direction. So some people say, well, maybe we need to design a gene therapy against the antisense transcript. That's an idea. And then some people are looking at actually sort of CRISPR, sort of DNA editing approaches to get rid of the genetic mutation. The most promising things are going directly at the root cause.
Okay.
Yeah.
So one more question related to that. How close are we to having effective gene-targeted therapies, such as ASOs, but you'll have to define ASOs, antisense, I can't say it.
Antisense oligonucleotides.
Thank you.
You were so close. Well, we do have some, which is exciting. So for tofersen, which is for the SOD1 genetic ALS, we have an approved therapy, tofersen. We are treating patients monthly with this. We did the phase 1 studies and the phase 2 studies, and it was very exciting because then it was approved and now we're offering it as therapy. And what we're seeing with this is that some patients get improvements in motor function. Many patients stabilize. And it seems to be the earlier you start treatment, the more likely it is to have a better response. So this was really game-changing. It's only 2% of ALS, but right. It said, hey, this is treatable. We can do this. Another program we're doing is that for other rare genetic mutations that are amenable to the same kind of therapy, personalized gene therapy. So there's a company called Eleanor, and they use the same kind of technology, this antisense oligonucleotide gene editing, and will come up with personalized ASOs for specific gene mutations. So this doesn't work for C9, it's only certain ones, but that we're about to do one of those. So that's, it's cool. We can use what we've learned in tofersen and potentially apply that to other genes. The real holy grail, and we don't know if this works yet or not, is in sporadic ALS, meaning the 90% of you that have ALS but you don't have a known genetic cause, can we modify a gene that is important in ALS and that could treat sporadic ALS? And that's exciting. There's several trials that are coming up with that mechanism. So,
well, that was the next question.
Yes.
Do you anticipate any possible impact for the sporadic ALS population based on the recent success in the genetic ALS?
Yes, I think we are hopeful that we could use gene therapy even in the sporadic population. Like I said, it's still to be determined, but One approach that a couple upcoming trials are targeting is there's this protein called TDP43, you've probably heard of in ALS, where almost all cases of ALS, I think like 98%, this protein gets kind of what's called mislocalized, meaning it goes to the wrong part of the cell and then it loses its function, or something happens where this TDP43 is playing a key role in ALS. And so this thought is, can we manipulate that TDP43 function to correct it, even in sporadic ALS, through a genetic approach? And we don't know yet, but that is something that we're hopeful about. So
we will keep our eyes on that,
and
for
what's
sure.
being asked in the lab and drug development right now. So I think there's a lot of progress that will be happening.
Okay. So as ALS is conducted, ALS research is being conducted worldwide. How interconnected is ALS research at Emory and among other ALS researchers?
Very, very integrated. And I would say within the US and also within, especially a lot of groups in Europe, there are a few, especially like the Netherlands, places that use a similar approach to collect blood, collect clinical data and genetic data from patients. So like our specific collaborations, we collaborate with, for example, the test that I was telling you about, where we're going to try and diagnose ALS from blood samples, or maybe diagnose isn't the right word, but triage. We're collaborating with NIH to validate that. We're part of a multi-center partnership with Answer ALS, which is several different academic centers, including MGH, Johns Hopkins, Emory. We collaborate with the Mayo Clinic a lot, especially on the autopsy studies and proteomic studies. Those are our main collaborators within the U.S. I think probably Mayo and Johns Hopkins are some of the closer collaborations. But we go to the same meetings. We work on this. It's essential to test what we find here elsewhere and vice versa.
So open data, or it's shared open data?
Oh, for sure. So all this that we talked about that you guys have given us in terms of your clinical information and your samples, that's been shared all over the world, so that a lot of different smart people are working on this. So we're very open about anyone who has a good scientific idea and would benefit from the resources we have, that's shared readily.
There are a lot of platforms. Of course, I mean, anything that is identifying, that's all wiped from the data, and then it's just the more generic information, age, male or female, things like that, disease duration, and here are the proteins. Because we can learn so much more from 10,000 data points than 1,000 data points.
Is artificial intelligence, or AI, assisting in ALS research in any way?
It's there. It is. And because of these large data sets, you need to be an intelligent user of AI. You need to. It's only going to be as smart as the information you give it and the question you ask of it. And how the data are organized can help you to use it more effectively. But we were talking about this yesterday. What AI platforms we like to use. It's made everything. It's made a lot of the analysis part easier, and it's also, I think, going to be really essential to understanding something that varies so much from individual to individual.
Oh, it's a good question. It's being used in so many different ways in ALS, and I think where we have this disease where it's a complex biology, we don't think in most cases there's a single cause, right? You probably need this risk gene and that risk gene and this environmental factor and that environmental factor, and there's variability. And so when you're looking at all of these different things, that you can't necessarily see anything with a human eye, but that's where when you take AI and say, do you see common threads here? Do you see a common genetic cause? Or there are multiple, biologies at play? Like, that's where AI can be really helpful. Lots of other things, like in technology too. I don't know, like with voice cloning, if any of you are familiar with that, but now AI can take any sample from your voice and create a synthetic voice that sounds like you. And it's kind of scary in other contexts, but it's very cool in ALS where people can bring back their voice and use it through technology. So that there's a lot of different ways. Yeah, go ahead.
The basis of that question
Yes.
is the president is tying together 19 FP64 NVIDIA chips
I've not heard about ALS inclusion in that. I don't pretend to know national policy, but I think we're always looking at, what AI is such a rapidly evolving field. We're definitely have it on our radar for what are the best tools we can use. But I think a lot of it is, it's only as good as the data you put in, whatever the platform is. So I think the good news is we have this really robust data set to put into whatever AI platform emerges as the best. So we're always on the lookout. But to answer your specific question, I'm not sure if ALS is included in that.
Okay.
And I think the AI increases the importance of the human component to this, because we actually need humans collecting the data, collecting the samples, measuring the proteins that we would input in any AI platform. So it hasn't replaced the need for humans, it's made them more important so that we can accelerate the research to meet the speed at which we can analyze it.
Keep your cards coming. We'll get your questions answered for sure. We don't have mics to go all the way around the room, so that's how we help facilitate that. I'm going to do a little more practical questions for a minute. Are you taking new patients, if you will, to take blood markers to test your theory?
Yes.
Yes. So if you would like to do that.
If you are giving blood in multidisciplinary clinic, then your blood could potentially be used for that. When we're selecting what plates and samples to run, we do kind of try and, if somebody's ALS is harder to diagnose, that patient would be one that we'd want to measure, because can we do better than what, than I guess the clinician in diagnosis, for example. It's the mystery cases and also people with other neuromuscular diseases that look like ALS. Those samples will be important.
Okay.
And we don't want to leave out our care partners either. We
Yes.
can always use members of
The family?
Control
Yeah.
essentially. If
Yeah.
you want to look at what's in ALS and what's not in ALS, you need people that don't have ALS. So
So that's a good point.
yeah.
All family members can participate. Since you've added 250 people since 2024, per the slide, is that a higher number of diagnosed cases than previously seen, or is that just a snapshot over the last several years?
It's been pretty steady in terms of the numbers that we've seen. So we haven't seen an increase in the number of people getting diagnosed with ALS, at least in Atlanta. I think the incidence of people living with ALS is going up as the population ages, and then as some things like respiratory interventions are keeping people living longer, you see more people living with ALS, but the diagnosis rate has not increased that we've seen.
Okay. Do you have any idea or theory why one person's ALS may progress slower or more rapidly that you would care to comment on?
No, I wish we knew that, right? I wish as a field we knew that. If you had to take one biomarker from the proteins we can measure in blood, neurofilament seems to track the best with progression, meaning higher is faster and then lower is slower. And most people think that that's probably just a side effect, because neurofilament's a protein in neurons, so if there's damage to neurons, you're going to see more of it. If you have more damage to neurons, you may see that would make sense that you would have more neurofilament. But it seems to track so well that some people are looking at, is this something that we need to understand more from a therapeutic standpoint? And there probably are other inflammatory proteins that we are now trying to measure on these assays that will track with progression in certain individuals. So I think that's another avenue to try and understand that.
So still something we're definitely exploring.
Nobody knows the
Absolutely.
answer. Nobody knows. A lot of ideas.
Would you say ALS is more prevalent in one particular country over another?
Generally, no. There's been these rare pockets, like there was this historical time that in Guam there were a lot of ALS cases, and they were wondering about an environmental factor. But in general, it's seen all over the world, similar incidents across geography and racial groups and things like that. So it seems to be pretty diffuse worldwide.
Okay. What percentage of ALS diagnoses are genetic? Now, I know what we quote. We quote I think it's 10 and 90, or
Yeah.
20 and 80.
So the 10% I think is accurate in terms of single gene in the way that we think of like genetic diseases, where mom or dad had this gene that gave them ALS and they passed it on 50-50 chance, and whoever gets that gene is at risk for getting ALS. A larger subset probably has what's called genetic risk factors, meaning that it's not one gene and you get ALS, but this gene increases risks a little bit, maybe that gene increases risk, and maybe this gene is protective, and you get some combination of many genes that could increase or decrease your risk for ALS. So we think that's at play at a large population of the ALS population, but still a lot to learn about that. It's hard when it's like a small contribution. You have to study huge numbers of people with ALS to find that. So I think that's a field that's really exploding. But
And genetic modifiers, why two people with the same mutation might progress at different rates. It's another.
Right. Okay. So what percentage of ALS patients would you say have FTD or frontal temporal dementia, and is that more common with genetic ALS versus sporadic?
Yeah, so about 10% of people with ALS will also have what's called frontotemporal dementia, which will cause cognitive and behavioral changes. That C9orf72 gene we were talking about is the most common cause of ALS and frontotemporal dementia together. But there are other genetic factors, and there's times where we don't find a gene, but there's still ALS and FTD.
Okay. What percentage of sporadic cases also include seizure activity? If you're aware.
I'm not aware. It's somebody, I've been asked that before, and it's not a known association, maybe that I could find.
Yeah, I don't think it's beyond the general population of who has seizures. We don't have ALS is not a known risk factor for epilepsy.
Okay. And can a cause for sporadic ALS be categorized as exposure to neurotoxins or infection?
It could be a small contributor, but so far there hasn't been a smoking gun, like smoking and lung cancer, where like this toxin is a clear cause of ALS. So I know this is a big area of interest, in particular in our military population, because veterans get ALS a little more commonly than civilians. But when they've tried to track it down to, is it this deployment or this toxin or this head injury, like it hasn't been one single thing that's been found to increase that risk. So probably at a small level is like one of the many contributors, but not anything that this one infection, this one toxin is a big contributor to ALS.
So this question was very similar. Do you think head injuries or environmental toxins have anything to do with ALS? So you answered about environmental toxins.
I think head injury is the same thing. There's been mixed studies finding mixed results. It's certainly not good for brain health overall, but not a really strong association with head injury and ALS in general.
Okay. Is spine and neck pain considered part of ALS? Or I guess, can you see spine and neck pain with that diagnosis?
I would say it's part of the like a secondary issue. So ALS itself doesn't affect the sensory nerves and trigger pain is a biological process. But you can imagine when mobility changes, when the muscles that support the head and neck and support posture get weaker, you can get secondary pain as a result. So we certainly do see head and neck pain, I think, secondary to the process of ALS, but it's more secondary to kind of the immobility and positioning and muscle weakness and that sort of thing.
Okay. Is there a common progression rate with how fast does bulbar onset ALS typically progress?
It's in general maybe a little faster, but there are a lot of exceptions to that from a research standpoint, but also in our database when I've looked at that say maybe it's on average a faster progression for bulbar patients, but there's some very slowly progressing bulbar patients. And same with maybe if it starts in the hands, maybe it's a little slower, progresses more slowly, except there are a lot of exceptions to that in the other direction. And it would be great to understand why.
Okay. Probably another secondary or related, does ALS directly or indirectly affect diabetes? Could it cause someone to become diabetic who previously had no issues?
Not a known association, so no. Typically the pancreas, the liver, the kidney, the heart, like none of those things are affected by ALS. So we do see, just because diabetes is so common, we certainly have people living with ALS that also have diabetes, but it's not a cause and effect.
Related to research, probably in the lab, are there animal models being used with ALS? Or do you want to speak a little bit about how?
Absolutely. I mean, one of the best mouse models for ALS was an SOD1 gene that's overexpressed in a mouse. So the first ALS causing mutation that was found was SOD1 in the 90s, and pretty soon after that, they were able to take that gene, overexpress the gene, make there be more of that protein in mice and in their motor system, and see a phenotype that looks like ALS in mice. There are many other mouse models now and other genes, and they're still used. They haven't been the game changer that I think people hoped they would be 20 years ago, thinking that, well, if you can cure the mice, you can cure the people. That just didn't happen that way. The mice are very different, and it takes one copy of this gene to cause ALS in a human. It took like 30 in a mouse, so that's already very different. But I think if we're going to develop a therapy or a treatment, of course we have to test this in model systems before we test it in people. We just also have to be very thoughtful about what we're measuring in mice and how we extrapolate that information back to humans. So I think it's still an essential part of research. It's just, it really cannot be the only approach.
Okay. One more question before we move on to maybe some conversation about drug therapies. So you mentioned about military veterans. Has there been any progress in establishing more than a correlation between military service and the 2 times frequency in veterans? Or is there anything you can speak to about that?
It is still an area of active research, so I think there's a lot of groups that are still working on this, but I think no, the short answer is we don't have that answer yet. I think we know enough to know it's not one thing. Like, it has to be multifactorial, because all of the is it just this one thing questions have been asked and looked at, and it's not. But I know, like, one study that's going on now, which is cool, is that the DoD has these samples of active military members who gave blood as part of their intake when they were healthy and all of that. And now we've been able to link who later got a diagnosis of ALS, and we have that blood from before they had symptoms. And so that's being studied to see, are there any markers before, like years before someone got ALS. So that's being studied right now. And so I think that's a really exciting approach and could lead to some of these answers, but it's still an active question.
That's new information to hear, so that's good. Okay, we're down to the last couple questions, so if you still think you have a question, go ahead and write that down. Related to drug therapy, are there compassionate use drugs available? How do we find out, or how do you go about getting them? Are they screened, or how do you qualify? So that's kind of a big question all in one.
Yes, it is. There's a few different pathways for this. So there are what's called expanded access programs, where a drug company has shown that their drug is safe, they're doing a clinical trial, and sometimes they'll offer this expanded access arm where they say, okay, if you don't qualify for the trial but you want to get this drug, then we have a pathway for that. And I'd say this is a very reputable, good way to get access to it. These programs are not that common, but if they exist, those are good. I will call it by name because I've been hearing a lot. There's one called Sphingogenics that is charging cash for participation, and the science and safety data doesn't really back that up. So I would say if they are asking you to pay to access it, then that raises some red flags about kind of scientific and ethical integrity. So I think if you go to clinicaltrials.gov or a trial finder, you can search expanded access programs for ALS. We don't currently have any enrolling at Emory, but I would say if you're able to find like a validated expanded access program where they're still monitoring safety, there's no charge to the patient, that is the most reputable way to access.
I think that's important for people to know kind of what their vetted ways are to do that on their own if they're researching that.
Yeah.
So that's helpful. What is your opinion of the tofersen therapy in sporadic ALS? We hear that Emory is involved in a trial.
Yes. So
Is that accurate?
Yeah, sure. I don't think anybody knows. That's why we're doing the trial. We know it's pretty safe from the SOD1 patients that receive tofersen. There are side effects, but we understand those because we've tried those. Things like myelopathy is the one that we know about the most. But it has a favorable safety profile, so it's a good thing to study. We don't know if it's going to work. So I think it'll be, I think it's an interesting trial. I'm glad we're doing it. And we all hope it works. But it's really sporadic ALS is not known to be associated with SOD1. So if somehow lowering SOD1 in sporadic ALS is beneficial, then we really need to go back to the biology to understand that better, because the mechanism is really still kind of being figured out for how that might work for sporadic ALS.
Yeah. No, and one thing I like that this trial is doing is when the tofersen trial was done for people with the SOD1 mutation, what they noticed was the neurofilament protein, you could see that going down before you could measure the clinical benefit. So it was this early marker of, hey, this drug is working before you could tell clinically. And so what they're doing in the trial is they're going to assess that neurofilament early and use it as kind of a indication of should we keep going or not. So that will minimize risk and kind of give us an early signal. And so I think we learned from tofersen and are applying that to how this trial is designed. And so it's good to know that each trial we do hopefully is a little better than the last one because we've learned everything we can from it.
Well, I think that concludes most of our questions. You all asked really great questions. And if we did not ask your question for some reason, or you've thought of one afterwards, you can email us and we will ask it through the doctors and get you all some answers. So you don't have to stop with that today. Thank you so much for sharing your time outside of the clinic and the patient room. We really appreciate it. We do have there are three multidisciplinary ALS clinics in Georgia, in Augusta, Macon, and here at Emory. But I would say that Emory has one of the most robust research program components for ALS attached to the clinic. And we're just so grateful to be partners with you all and to be able to be in partnership. So thank you. I think Dr. Thomas and Dr. Fournier will be around for a few minutes afterwards if you have some questions. Our community partners are still here if you didn't get a chance to see a certain table or two. It's really our privilege to be able to serve you, and we hope that you have learned some new information, maybe had some meaningful connections, and reassurance that we're here to walk alongside of you. We do have, typically, if you've been here before, we have paper surveys. We're moving digital a little bit more. So if you would take a moment to scan this QR code, there's just a few questions we have for you about today's program. This is for you all. So we want to provide information and resources that benefit you all. So if you have feedback for us, we'd greatly appreciate it. But this concludes the formal part of our program today. Thank you so much, and we look forward to seeing you soon and hopefully at our 25th anniversary walk in September.